Dear students, in this video we are going to discuss about production of gametes. Gametogenesis is the formation of gametes in gonads that takes place in testes in male and ovaries in female. Gametes are formed from primordial germ cells of gonads. It includes spermatogenesis and oogenesis. Spermatogenesis for sperm production and oogenesis for womb production. Gametes differ from somatic cells in number of chromosomes. It is in diploid one, 2n chromosomes. Meiosis plays a major role in gametogenesis. So there are the three stages of gametogenesis. One is multiplication phase, growth phase and maturation phase. So the first one is spermatogenesis. The process of sperm formation occur in the seminiferous tubules of testes. At onset of puberty, hypothalamus secrete gonadotrophin releasing hormone GnRH. GnRH initiates secretion of follicle stimulating hormone which induces spermatogenesis. So there are two stages of spermatogenesis: formation of spermatids and spermatogenesis. Formation of spermatids. During development, the primordial germ cells migrate into testes and become immature germ cell called sperm mother cell. So this sperm mother cell is present in the inner surface of the seminiferous tubules, and then in that place germinal epithelium cells initiate this spermatogenesis process. So the first step of this um, phase is multiplication phase. At sexual maturity, the undifferentiated primordial germ cells, that is diploid in number, divide several times by mitosis to produce large number of spermatogonia. And then next phase is growth phase. Some spermatogonia stop dividing. They actively grows into larger primary spermatocyte by obtaining the nourishment from Sertoli cells. So the Sertoli cells provide the nutrition for the growth of this spermatogonia. And then developing sperms are embedded in the cytoplasm of Sertoli cells. So the sperms, the growing sperms were present in the cytoplasm of the Sertoli cells. Then the last phase is maturation phase. Each primary spermatocyte undergoes two successive meiotic division called maturation division. In meiosis 1, two haploid cells called secondary spermatocytes N are formed. In meiosis 2, four haploid spermatids are formed. And then after the spermatid formation, now the spermatid get active matured one. So the spermatids are spermiogenesis process will takes place. Spermatids are non-functional and non-motile, but and also considered as a inactive one. These spermatids get transferred into active spermatozoa by a process called spermiogenesis. Spermatid's head remains attached to Sertoli cells, tail hanging in lumen of seminiferous tubules. During spermiogenesis, spermatid's length increase. During the process of the spermiogenesis, spermatid's length increase, centrioles are rearranged, mitochondria are coiled, and acrosome, the head portion, is formed. So this is the pictorial representation for spermatogenesis. The initial stages, that is immature stage, spermatogonia in diploid chromosomes, that is 2n. After the mitotic division, after the completion of mitotic division, primary spermatocyte is formed. It is also diploid in number, and then it can be divided into two, that is n haploid chromosomes were divided through the meiosis 1. and then secondary spermatocyte is formed. This secondary spermatocytes are divided into four spermatid with the help of meiosis 2 cell division. And then spermiogenesis process will takes place. Then this spermatid, that is inactive spermatid, is converted to active spermatozoa, that is mature spermatozoa will formed. Spermiation. After spermiogenesis, sperm head became embedded in the Sertoli cell and finally released from the seminiferous tubules by the process is called spermiation. And then next one is oogenesis. The process of formation of mature haploid female gamete is called oogenesis. The female gamete produced is called as ovum or egg. Oogenesis differ from spermatogenesis in several ways. Oogenesis is different from spermatogenesis. Oogenesis occurs only in the ovaries. Every month, each ovary takes an alternative turn in releasing an egg. In unilateral oophorectomy, the single ovary releases egg. So this is a pictorial representation for oogenesis and then follicle development. So the first immature oogonium, it consists of two end, that is diploid. It can be divided by mitosis and then primary oocyte is formed. This primary oocyte, it is also a diploid in chromosomes. This primary oocyte, after the divided into a small oocyte and then larger oocyte. So this larger oocyte is considered as a secondary oocyte. This process takes place with the help of meiosis 1. Again, this secondary oocyte get activated. This secondary oocyte is active form when the sperm will attach the ovum, that is oocyte. Now the fertilization process takes place and the zygote will form. In the follicular development stage, there are primordial follicle, primary follicle, Graafian follicle, that is mature follicle, and then Graafian follicle. Primordial follicle, that is the initial follicle. Primordial follicle are before birth. Then after birth, primary follicle will form because of the nutrition. And then this primary follicle, again it can be growing bigger in size, that is growing follicle, and then the next stage is maturation. So the follicles get matured and the ovulation get happened and then corpus luteum will released. So stages of follicular development, there are 5 stages of follicular development: primordial follicle, primary follicle, secondary follicle, tertiary follicle, and Graafian follicle. So the first phase of this oogonia is multiplication phase. Oogenesis process begins inside the fetus before birth. So the oogenesis process happens inside the fetus before birth. During the 6 to 8 week of female embryonic development, primordial germ cells grow and begin to differentiate into oogonia. So the primordial germ cell is considered as a mother cell for both male and then female, but depends on the sex determination. It can be differentiated into spermatogonia and oogonia. They divide by mitosis and produce large number of egg mother cells or oogonia. Millions of gamete mother cells are formed within each ovary. This happens between 3 to 8 months of gestation in the embryo. No more oogonia are formed or added after the birth. So the oogonia are present inside the present only in the fetus condition. So it does not synthesized, secreted or matured during the after the birth. Okay. And then next one is growth phase. Some oogonia stop dividing, begin to grow in size and form primary oocyte. Cellular organelles like endoplasmic reticulum, Golgi body, mitochondria are developed during this stage. They enter into the prophase 1 diploidy of meiosis and get temporarily arrested at this stage. Primary oocyte do not divide. further until the female became sexually mature. So the primary oocytes are present in the ovaries, but it cannot divide. It can divide only after the puberty. The primary oocyte then gets surrounded by a single layer of granular cell to form a primordial or primary follicle. And then the last phase is, that is maturation phase. At puberty, due to induction of FSH-LH, that is follicular stimulating hormone, and then luteinizing hormone. At puberty, due to induction of FSH and LH, primordial follicle grow into primary, secondary, and tertiary follicles. In meiosis 1, secondary oocytes and polar bodies are formed. In secondary oocytes retains bulk of the nutrition-rich cytoplasm. In meiosis 1, secondary oocytes and polar bodies are formed. In secondary oocytes retains bulk of nutrition-rich cytoplasm. Secondary oocytes precedes meiosis 2 only up to metaphase 2. Its division is arrested in this stage. Thank you.